So it gives me great pleasure to introduce our next speaker, who's Professor Troy Lund. He works at the Leukodystrophy Centre at the University of Minnesota, and it's great to see him here again. He joined us for the first time last year and does amazing work. And he's going to do a deep dive into adrenoleukodystrophy. Thanks, Troy.
All right, thank you very much for the invitation. So this is going to be a brief talk about a new project. We call it Operation Deep Dive, informally, and I'll show you why. So in our adrenoleukodystrophy clinic, we've been seeing families for over 20 years, and historically, these families would be coming in because of a new diagnosis, because the child presented with some kind of severe symptom, sometimes as a seizure, sometimes it was an abnormal MRI after a head injury, sometimes it was behavioral changes that led to an MRI, or once in a while there would be a family history of ALD and cerebral ALD in the family, and so the next child would be born, the next boy would be born, screen positive, and we'd see that family. But enter now the realm of newborn screening, which is carried out in the majority of the states, United States. We've identified many, many more boys with elevations in very long chain fatty acids and subsequent variants or mutations found in the causative gene ABCD1. And so inevitably, these families will find the centers with expertise that are scattered around the country and get referrals and make appointments and show up. So instead of my clinic filled with 5, 6, 7, 10-year-olds with ALD, a lot of it is now babies. A lot of newborns, a lot of families just coming, not just to talk about bone marrow transplant, really just to get educated about the disease. And so I have this clinic full of babies. And the first question, after the parents go online, they read about ALD, they often try to learn about cerebral form, which is very scary to them. They learn about AMN, which is very confusing to them. They learn about women with ALD, so on and so forth. But the most common question is, will my child get cerebral disease? And so the answer is just a statistical number. 35 to 40% is what's in the literature. It's what we all write about in the first two sentences of every paper we write, that it's about a third or up to 40% will get cerebral disease. And that's rather unsatisfying because they'll say, well, what about my child? I may or may not have a family history. Does that matter? Which 35 or 40% is it going to be? Hard to know. We've certainly seen families of boys where you have 4 boys and 2 may be affected, 1 may be affected. All the boys carry the same mutation. It's a big part of the ALD we don't understand. But at the end of the day, it's not very satisfying just to say, well, I don't know, it's 35 to 40%. So what's some of the background on the newborn screening? It all started in New York in 2013. They were the first state to start newborn screening for ALD. In 2016, ALD was added to the RUSP panel, which is our national guidance on whether or not states should screen. And it helps us get screening passed at the statewide level. And then in my own state, Minnesota, we started screening for ALD in 2017, and since we've been screening since. So what's been very interesting is that we've come across a lot of families, again, with young children, babies that come to clinic, with no family history, and they get their genetic report, and it says variant of unknown significance. And trying to define that is actually very difficult, even if you're an expert in the field or a geneticist, and families get very confused by it. And Stefan Kemp and I wrote a paper 2 years ago now about what are these variants, which normally stands for variant of uncertain significance, or for us, very unclear situation, but also for families. And many of the families I'm seeing, they have zero family history backwards through many generations. And many of them come in with well-documented family trees. Furthermore, a lot of them have had testing through the family before they get to me, and they went back and tested all the males. The males have been shown to carry the variant, but there's no symptomatology at all. And so one of the things was, well, maybe some of these variants have different implications for the type of phenotype, which is not the dogma. Generally, we think about this rate of cerebral disease at 35 to 40% across all the variants or mutations, which, as Florian showed, there's over 1,300 now. But perhaps, as there is with many other diseases, that some of the variants may have a different risk for cerebral ALD, in fact, a very low risk. And so that's where this project was born out of. So as Stefan and I published, is just a summary of what these variants of uncertain significance, what their rate is. If you look in the ALD database, or the ABCD1 variant database, historically, the rate of VUS reporting was about 20% or 21%, versus 45% of the variants being called pathogenic. If you look at the recent data we collected from the newborn screening, that VUS rate went up 200% to 62%, while the pathogenic rate went down to 29%. And I can show you what this looks like in a pie graph because it's much more striking. So in gray, we have the variants of uncertain significance at 21%. But now from newborn screening, you can see over half of the pie now is the variants of uncertain significance. And again, these are boys that are picked up. They have elevations of very long chain fatty acids that are above the cutoff for normal, and they come back with some kind of a variant or mutation in the ABCD1 gene. And so just briefly about variants of uncertain significance, that again, many of these have been identified since newborn screening has started, and it's unclear whether or not these VUSs are connected to a health condition. That's why they're a variant of uncertain significance. Often they're rare in the population, so we really don't know much about them. And of course, getting more information about them will help us understand which variants may be significant and which aren't. A lot of the data actually comes from population data, which you can find online, or you can go through literature and try to find individual papers about each variant. And then tracing variants within families is very important for studies like this. There's myself and the good Dr. Kemp, very excited about talking about variants of uncertain significance. This is back in 2017. I should point out that Dr. Kemp maintains the ABCD1 variant database, which is incredibly helpful to this. And that database is a community-driven project. And so anytime I get a new variant, or if I have a child who either gets adrenal insufficiency, cerebral disease, a man with AMN, or women who gets any symptoms, I report it back to Dr. Kemp, and so we can collect more information about these variants. So we recognize this might be an issue even 5-6 years ago when we reported the implementation of state screening from Minnesota about our first few years of experience screening for ALD. And in the abstract of this paper, interestingly, we call out that there are just milder cases that might be underrepresented. Other challenges include a high frequency of variants of uncertain significance in ABCD1 and an inability to predict phenotype severity. So we posit through thoughtful planning to address these novel challenges, and coordination by dedicated specialists that'll be imperative for successful implementation of population-based screening for XALD. And an interesting phenomenon in Minnesota is that we're a low birth rate state. And so, but we have some very large families that have lots of children, and it just so happens that some of these families carry variants of uncertain significance. And so we have an extremely high proportion of children in clinic that come in with this dilemma, is that these long family trees. They have many people now in the family identified with the variant, but they absolutely have no adrenal insufficiency, no cerebral disease, and no AMN. And so that posed a challenge of what to say and what to do. So how can we figure out what all these variants do? There's at least 1,300 variants. Any variant associated with a person that has symptoms like AI or cerebral disease is no longer a variant of uncertain significance. It is a disease-causing variant. You can just put that one away. The rest of them, we have to figure out what to do. So you could actually design experiments in the lab and perhaps test every variant in some kind of fancy cell culture experiment. You could devise some kind of transplant experiment or some kind of animal experiment. You could test all the experiments in a mouse. You could maybe do some computerized modeling, some in silico modeling, and get some predictions about the variants, whether they change protein structure, function, lead to shortened half-life, etc. But there are drawbacks to each of these things. Cell culture doesn't always mimic human biology. It doesn't look like the human body. It's fairly artificial. For mice, we'll have the various mouse models. They really don't get cerebral disease, and they don't suffer from adrenal insufficiency. So they're not the ideal model for this. And then as far as computer modeling, well, computers are only as smart as the data put into them, and if we don't have a lot of robust data, then the modeling's not going to be very good. So, and then I'll just bring up a project adjacent to this that we call the Gray Zone, which ALD Connect funded in 2022. And the Gray Zone has a lot to do with these variants. And one of the central questions within the Gray Zone is the ability to separate these variants based on a very sensitive and newer biochemical moiety called lyso-PC, which is very sensitive. We find it in high levels in persons with ALD. And one of the questions is maybe that marker will be able to separate some of these patients. Some of the criteria we discuss about enrolling patients in the Gray Zone is the babies that are identified via newborn screening, because they most often have a VUS. Is the ABCD1 mutation a VUS actually? Is everybody in the family with ABCD1 mutation asymptomatic? Because again, if you have symptoms and you have a variant, then it's not a VUS. It is then a disease-causing variant, and you can put it to the side. And again, this is in collaboration with Stefan Kemp, who I should try to name on every slide. So another way to figure this out is statistically. So just a numbers game, which most of population genetics is just a numbers game and statistics. So if I had a child with a VUS, a boy, and I had 1,000 men in my clinic with the exact same VUS, and I saw no AI, no adrenal insufficiency, no cerebral disease, and no AM, no AMN symptoms, I would say that variant is probably benign, and that it is not disease-causing, and in fact, it really doesn't lead to ALD at all, despite elevations of very long-chain fatty acids that are above the normal range and a mutation in ABCD1. statistically, if you saw 1,000 individuals that were asymptomatic, odds are that that variant has nothing to do with the disease. In fact, you could lower this down to 100 men. So if you saw 100 men with no symptoms, no AI, no cerebral disease, and again, you'd expect dogmatically 30 to 40% of individuals with elevated very long chains and an ABCD1 mutation to get cerebral disease. If you didn't see that across 100 individuals, and again, you had no AMN symptoms across these individuals, you can probably dismiss this variant as disease-causing and is probably not, in fact, associated with ALD. And so you can model this type of, you can model the answer to this question statistically through mathematics. And this can go on and so forth, and we won't keep dividing down because you eventually get to 1, which doesn't help. So that leads us into this new research study, is this Operation Deep Dive. And so we've picked various variants to focus on. This is an example of one of them. This is valine 583 methionine. It's one of the ones we're interested in, and it's a very common one in Minnesota, which sparked our interest initially. And we've interviewed dozens of patients, collected dozens of samples, and have a case study that is nearly complete now about this variant. So why this one? Well, again, we described it in Minnesota families, and these families are very large. There are, in fact, 4 or 5 families that are all connected. The families have up to 20 individuals in each family that we've spoke with or identified in the tree. It makes it ideal to have enough numbers to power the study. Many of the people, i.e. many of the men in the family, are reachable and can be examined and samples collected. So just as an example of what kind of data one collects on this, and again, this is just a sub-cohort from a family, as that we get we're focusing really on the males. We collect the age, whether they have AI, whether there's any cerebral disease, and again, whether they have a diagnosis of AMN or symptoms of AMN. And you can see there's a child who's age 4, no AI, no cerebral disease, but again, have to discount that because maybe that child has not developed cerebral disease. But that child's grandfather, who's 74, also has no AI, no cerebral disease, no AMN, and is a retired farmer. There's another male who's age 5, again, no symptomatology. There's another great uncle, age 74, no symptoms. There's another uncle who's 49, who's a banker, no symptoms. This guy here, he is a triathlon, carries the variant, no symptoms, no nothing. And you go through the family tree and you start identifying all these males, and first you get them tested, they have the variant, but nobody in this part of the tree has any symptoms and no AI. In fact, through all the connected trees with the same variant, you can collect dozens and dozens of men with 0 symptoms and 0 AI. And so this is just an example of what we're trying to do. So the next question, well, what about the very long chains? Where are they falling? And this goes back to the Gray Zone Project to some extent as well, is that, so this is a graph of C26 levels, and the graph is from Kennedy-Krieger, and this is the levels they report for normal individuals, persons with ALD, this would be the median and 1 standard deviation, and this is ALD girls. And so we're trying to collect as much data on men with these variants as we can. And this is where they fall. So they're above the normal range for sure, but they're generally below the ALD range, which makes it confusing, save for one individual, which I'm actually going to retest because this person is so far out from the rest of the family that I think it might be erroneous. And so this then enters into the Gray Zone Project is how do you separate these individuals? And so perhaps through Dr. Kemp's lab and his testing for lyso-PC, we'll be able to separate some of these individuals out. But this is where their very long chain fatty acids fall. So what are the next steps? So I try to track down as many male family members with this particular variant as an example, valine 583 met, present them with the research opportunity. Many of these families turn out to be in Nebraska, Minnesota, and Illinois. So but we're always looking for more. And so then what's involved when we're trying to nail this down? Ideally, a clinic evaluation, an evaluation by an expert in ALD, an evaluation by a neurologist, blood draw for plasma is helpful, cheek swab for DNA is also helpful if we need to yet verify the variant. So then after we collect all this data, we will statistically model based on population studies and based on the numbers of men and boys we collect within the study for cerebral disease. We can do this across, actually, VUSs to bolster the power of numbers because the statistical modeling requires large numbers of individuals. And so those that we deep dive on, we can do that on. And this is not a difficult concept because in many diseases, there's not just one risk stratification. In fact, if you look at colon cancer, we risk stratify people based on family history. So why wouldn't ALD be any different? That there could be different levels of risk. There might be high-risk variants, there might be low-risk variants, and I would argue there might be no-risk variants. And so that's what we're trying to get at here. And then this allows us to actually provide individualized monitoring for families. You know, the dogmatic guidelines that we've published that we adhere to is that generally, if you have a variant in elevated very long chains, you are getting MRIs starting at about age 2, every 6 months through age 12, and then yearly thereafter. Then blood draws periodically to look for endocrine function. And so maybe by being able to risk stratify some of these variants, we can actually modify the way we do surveillance and therefore creating an individualized approach to every family. We have an ALD national registry where we follow multiple families around the country. Unfortunately, I can only consent families in the United States, so it really doesn't apply to showing this internationally, because that's what our consent form allows us to do. But certainly if there are questions or you think you might have a variant of interest or unknown significance, I'm always happy to answer by email. Thank you.